Thank you, Imran, and thank you, the organisers, for arranging this meeting today. It's been fantastic. Sam and I, hello Sam, if you're there online, I know, we thought it would be a nice occasion to say a few words about Mike Thorndyke. As many of you all know, Mike was my PhD supervisor many years ago now, but sadly Mike passed away earlier this year. So we thought this would be a nice occasion. Mike would have loved to have come to this meeting. So Mike, first slide. Michael Charles Thorndyke was born on the 22nd of October, 1946 in Walthamstow, East London. He was the eldest son of Alfred and Doris Thorndyke. And I thought it'd be nice to read out some words from Chris Thorndyke, Mike's younger brother. He kindly sent these to me while I was trying to prepare an obituary for Mike, which was published recently. So Chris says, he was always fascinated by wildlife, particularly those living in sea and fresh water. As a young boy, he used to go fishing with a rod made out of an old army tank aerial, which was common apparently in the 1950s. At our family holidays in Dovercourt on the East Coast, he was always rummaging around rock pools and seaweed-covered rocks, collecting various creatures to study. When we stayed in Colwyn Bay in North Wales, we would frequently go on long hikes, and Mike would have a number of glass jars in his rucksack to collect bugs. When we got back to the guest house, he would drip some ethyl alcohol on a piece of cotton wool and drop it into the jar to store the bugs. So it wasn't really any surprise that Mike would go on to study zoology at Queen Mary, which was the nearest academic university close to where he lived in East London, and of course where I work now. So he studied for a BSc in zoology from 1965 to 1968 and was awarded first class special honours, and then proceeded on to do a PhD under the supervision of Alan Thorpe from 1968 to 1971. And the picture below the Queen Mary's building is actually Mike's thesis, which I inherited from Alan. When Alan retired, he passed on his thesis to me, so it's in my office if you ever want to look at Mike's thesis. And in the top right, you can see one of the many beautiful electron micrographs from Mike's thesis. He studied the endostyle of protochordates. We don't use that term so much now, but urochordates particularly. And the main reason for doing so emanated from the work of Alan Thorpe and also Barrington, Thorpe's supervisor, who was also involved in supervising Mike's work, because there's this interest in the endostyle as a sort of evolutionary precursor of the thyroid gland. So it was an endocrinological project, but with an ultrastructural component, and it was published 50 years ago. I noticed, here we are, 50 years ago, Mike's first paper was published in the Journal of General and Comparative Endocrinology. Mike then took up a number of short-term lectureship positions, but finally found a home at Bedford College in Regent's Park, part of the University of London, and was there for 10 years, where he lectured in physiology and endocrinology. And here's a wonderful photograph of Mike looking like the mad professor. I'm not quite sure what he was doing there, but he's got some solvents of some sort. He carried on his research on urochordates. And one of the highlights published in Nature in 1978 was the, at that time, quite innovative use of immunohistochemistry to visualize proteins in invertebrates. And he particularly showed that using antibodies to thyroglobulin, you could detect thyroglobulin-like proteins in the endostyle, providing further evidence of this relationship between the endostyle and the thyroid gland. In 1985, Bedford College merged with Royal Holloway, and he moved along with all the other zoologists at Bedford to that campus. And actually, it was in 1985 that I took up a position of being an undergraduate student at Royal Holloway and doing a biology degree. And Mike taught me physiology in the first year. And then in the second year, I took his comparative endocrinology course. And one of the things he taught us was about lots of different peptide hormones that he had been working on from a comparative perspective. And this was actually a paper that was published at the end of 1986, which was one of a series of papers reporting evidence of cholecystokine in gastrin-type neuropeptides in Ciona intestinalis, both functional and anatomical and biochemical studies. And so that's where I first learned about these hormones that we now all work on. And by pure coincidence, the following January, I was also doing an invertebrate zoology course, and that is actually my drawing, my first ever drawing of a starfish. I kept my notes. We did a section every week, and the one we did in January was Asterius rubens. And I was always fascinated by the beautiful symmetry of the animal. And I think I actually wasn't very good at dissecting, really, to be honest. And it was what was so nice was if you messed up one arm, you got four more. So I quite liked starfish. And so in that spring of 1987, I basically stuck together the idea that I would take Mike's idea of studying these gastrin and cholecystokinin type hormones from an evolutionary perspective and just extend it to echinoderms, because I like these animals. And so that was the idea I went to Mike with as an undergraduate student, as a possible third-year research project. He thought it was a good idea, and so we had a go. But the results were basically negative, and that's why there's a blank space there. The antibodies that we used to cholecystokinin did not stain anything in the starfish nervous system. And unfortunately, we then had to wait, as it was, I think, about 34 years until last year, when finally our group succeeded in discovering the actual cholecystokinin, that they are present, it's just the antibodies didn't cross-react with them. And we now have functionally characterized these peptides in some detail in starfish. But Mike was always ready for these sorts of situations. And so when we had this rather disappointing negative result, he said, well, I've got plenty of other antibodies in the freezer. Why don't you try some of those? So that's exactly what I did. And thankfully, one of them did actually give us some beautiful staining. And Wenbin, you'll recognize hyperneural region of the nadal nerve cord there, some beautiful staining of the hyperneural neurons with the antibody to a molluscan peptide from murfamide. And this was the first study to show anatomically the presence of neuropeptides in the echinoderm nervous system. And it was published in 1989. And it led on to me being able to go to Florida. Mike was always very well connected, and he had already was already collaborating and had contacts with Dave Price and Mike Greenberg, who had discovered murfamide originally in mollusks. And he enabled me to go out to Florida, which was very nice, in 1988 and 89. And there I succeeded in purifying these peptides that we now know as sulfamides, S1 and S2. And here are some pictures from that time. That's me and Mike at the Third International Congress of Comparative Physiology and Biochemistry in Tokyo. At that time, you could get to go to one international meeting, and I chose the one that was as far away as possible from England, and managed to go to Tokyo, and never been back since. Actually, I must go sometime. And that's actually the day of my PhD viva. After the viva, if I'm looking a bit tired, that's why I'm sitting next to Mike, celebrating after the viva in Mike's office. And earlier this year, we actually, to bring this full circle, we actually managed to finally discover the receptors that these sulfamides interact with. And not surprisingly, I decided to dedicate the paper to Mike in his memory. So that's, I think that takes us up to 2002. And I'm now going to hand over to Sam, who I hope is there online.
I am.
Sam? Very
yes.
good. So I will do the slides and you do the talking. I'll forward the slides when you want me to. Over to you.
Thank you so much. So thanks everybody for staying. I think Mike would have hated being between you and the pub, so we're going to keep that nice and sweet. But yeah, so I've met Mike the first time, I think in 2000. It was in an Echinoderm conference, an international one that was held in New Zealand. And I remember having an amazing night talking with him and having a lot of beers. And I think that's probably the reason why he hired me a couple of years later when I was looking for a postdoc. I think we had a connection, and that's really how Mike was working. It was creating personal connection and seeing things in people. But when the 21st century started, Mike took a completely different challenge. So he was an amazing scientist, extremely well connected, and he decided to move to a place he really liked. He visited many times over the years. Was this marine station that you can see on the picture. So that's the Kristineberg Marine Station in Sweden. So this place is the third oldest marine station in the world. It was founded in 1877. And at the time, it was kind of still a little bit old-fashioned marine station. And Mike took the position there as director for the Academy of Science and also for the University of Gothenburg a little bit later, and really made the deal to try to take the station into the 21st century. So he negotiated with his position the possibility to really start like molecular biology there. So he made a molecular lab. He really introduced the omic revolution inside the station. And since then, it became one of the most visited marine stations in the world. And he also developed different model species. For example, we had this talk a little bit earlier about phylogeny, and actually Mike promoted the use of Xenoturbella, this funky worm living in the mud around the station. And you have still every year people visiting from all around the world to use them and try to resolve the mystery of that creature. But there was also an echinoderm that became really close to his work, and that's Amphira filiformis, one of the brittle star that is really common in the vicinity of Christineberg, and it really was one of the person who introduced it as a model, and many people in the room, I'm sure, are really familiar with that organism. That is, we have a genome project on the way, and a lot of work done, for example, by Paola Oliveri, that I heard is in the room. So can go to the next slide, please. But Mike didn't want to just promote the Christineberg Marine Station, and he took the huge challenge to try to lift marine science in general in Europe, and he decided to promote the use of marine stations. So he created all these networks to try to promote marine science and marine stations. So he was, for example, the coordinator of the Assemble program, a European project that's with the goal to offer opportunities for researchers to travel to different marine stations to do research. And the station in Christineberg was absolutely amazing because it was the one of the most visited one. He was also the coordinator of the EMBRC, the European Marine Biological Resource Center, and both this initiative, Assemble and EMBRC, are still alive today and still quite active and really promoting the marine science. So Mike was really a connector. He had this amazing network. I never met anyone who didn't like him, and he knew so many people that he was really good at promoting science, promoting marine station, and so on. And one of the title he was the most proud of is he was the president of Mars. And I remember him introducing himself like, "Hello, I'm Mike Thorndyke. I'm the president of Mars," and that doesn't mean I'm crazy. I think that was one of these jokes, but that was also showing the both side of Mike, that he was extremely serious, extremely committed, extremely generous with his time, but also a little bit goofy sometimes. Next slide, please. So because of all these commitments he had now in science policy and so on, he didn't really have the time to be in the lab anymore, but he still was very interested in science. So basically what he did was to create a fantastic environment for research in this marine station. He basically developed new approaches, like I mentioned, omics, but then he also offered opportunities. And I have to say that I'm one of the lucky ones. When he hired me, I arrived there as a young postdoc, and he told me, do whatever you want and don't worry too much about money, which is the best postdoc you can imagine. So I arrived there and I started to work at the beginning with him on stem cells, on regeneration in echinoderms. But then because he was so good at inviting people, we had the visit at some point of Lloyd Peck from Bath, who was just having a look around. And at the time I was culturing brittle star larvae. And I remember Lloyd saying, "Oh, you can do that. That's really great. I think you should maybe look at this thing called ocean acidification." And neither Mike or myself ever heard about this, but we thought, oh, that looks interesting. And we started to make new experiments about that. And that became a really big issue. Now you have more than 10,000 publications that were published over the last 10 years. But thanks to Lloyd and the vision of Mike, we basically started working on that subject at the very beginning of the field. And since then, we also promoted best practices, developed infrastructure at the station in Christineberg, and became really leader in the field of acidification. Together with many colleagues, we published more than 200 publications in the field, contributed to best practices, contributed to policy, like the work Mike and I did were used for the IPCC report that was published this year, for example. And we published a lot of really cool methods, like new methods to measure pH inside sea urchin larvae and things like that. So it really created an environment for really high-level science. Next slide, please. But the reason why I think it was important personally to have this talk today is because Mike was much more than just a great scientist, a connector, a really interesting person. He was really a true leader and an inspiration. He's the kind of guy you want to be. He was really likable, really funny, extremely smart, extremely generous. And I was reading this book, Thinking Fast and Slow, that I really recommend if you've never read it. And the author is describing a relationship he had with one of his colleagues. And that's exactly the feeling I had with Mike. He said, we enjoyed working together. He was always very funny. And in his presence, I became funny as well. So we spent hours of solid work in continuous amusement. The pleasure we found in working together made us exceptionally patient. It is much easier to strive for perfection when you are never bored. We were sufficiently similar to understand each other easily, and sufficiently different to surprise each other. The work we did together during those years was the best either of us ever did. And I think that was totally true with me. Mike was a catalyzer for the work I've done. Next, please. But even after his retirement, when he moved to the Netherlands, he continued to work, and he was extremely visionary. For example, he joined this meeting in where there was like the beginning of the introduction of the field of ocean and human health in Europe. And he really saw the potential of that. And he was at the time editor-in-chief of GMBA UK, and he edited a special issue on the subject. And today, ocean and human health is one of the main filter that the scientists are using to prioritize the science of the future. So that's one of the filter that is used, for example, for the UN Decade of Ocean Science today. So he really had the vision of what would be important in the future. Next slide, please. And just to show how generous he was, again, during his retirement, he traveled many times to the Bahamas to work with this research center, the Center for Ocean Research and Education, to work with a guy you see on the picture holding a turtle, Owen. And Mike went there multiple times to teach, to collect samples for colleagues and so on. And more recently, he even found some funds to basically do experiments there, which is extremely challenging, but he succeeded in collecting really interesting data on the impact of ocean acidification on seafood. And unfortunately, he won't be able to write a paper, but Owen and myself are dedicated to do that. So there will be at least one more paper by Mike in the near future. So thank you. I'm going to leave the last words to Maurice. Thanks, everyone.
Thank you, Sam. So yeah, I just thought, I mentioned that Sam and I have written an obituary that was published in JMBA just quite recently. And I thought it'd be appropriate to just read the last actual paragraph of the obituary. The photograph is, I thought I'd also have a photograph to show you. I love this photograph. This is me with Mike. And my son, who was then 2, he's now 20 and taller than me and bigger than me. But I thought this would be sort of symbolic, really, of the way Mike was. He was just like became a real friend to me, and likewise Sam and many others. So Mike was friendly, kind, generous, humorous, and humble. And it was these characteristics, together with his infectious enthusiasm and energy, that made Mike so effective in uniting and mobilizing people to accomplish objectives internationally. Accordingly, Mike's many important contributions to marine sciences will surely have long-lasting impact. Mike was also a loving husband, father, grandfather, who had many interests outside of science, perhaps most especially a passion for music in all its forms. It was for us an honor, as for so many others, to be Mike's colleague and friend. Thank you. And I think one or two members of Mike's family, I think, are online listening in. So hello to you all if you're there, and thank you, everyone.